our meeting here. I do have Ana Maria Vasquez Catoni of the Holland Lab is still collecting more responses for that survey she has. So this is a grower survey for, I believe, Wisconsin cranberry operators. Ana Maria, text me if it's actually all cranberry operators, but I believe if you're a Wisconsin operator and you haven't filled that out yet, go ahead and capture that QR code. I'm gonna, hopefully I can advance through our slides here. Yeah, so this is the June 28th, 2024, the June Cranberry Brown Bag. We have some pretty exciting talks. Like I said, we've been looking forward to this water and nutrient uptake into berries speech from Amaya, and that's going to be 30 minutes of our content today. Then we're going to have Q&A for Amaya as she will head out to give one of her students is doing their thesis presentation, and then Pam is going to give us a field update. So I want to take a minute to let Grant give us our quick welcome, and then we'll dive in, I believe, to Amaya's presentation.
Hey, everybody. I just want to thank you for coming to another one of our brown bags. I did do a welcome on our previous brown bag when I was in it for about 2 days as the new executive director, and we're nearing day 40 something at this point in time. And from touring some of the farms and seeing some of the people, I sincerely hope that the weather is treating you well. And if it isn't, I hope that research like this and research that's coming out helps to make your lives a little bit easier. I love the topic today. Anything having to do with water, whether it's internal or external, should be a focus for us for the next few years. And with that, I'm going to turn it over to the folks that know a lot more than I do and just say thank you and also welcome.
Fantastic. Thank you, Grant. We will hop in to Amaya. I assume, do you want to share your screen here?
Yes, I will share my screen.
I'll stop mine.
I'll put these on. Sorry, I'm a little bit dispersed. I was in another meeting right now, so I'm just like trying to get.
It is a busy week or a busy month for growers. It is also a very busy week in the academic world.
All right, so hopefully there you can see it on presentation mode. Is that good?
Yes, and I just want to remind everyone, we do have closed captions on. If you have a question, you can either unmute yourself and ask, or you can type a question into the chat. If you want to ask an anonymous question, use your chat drop down to say, ask the question to Allison, and I'll ask the question anonymously on your behalf. So with that, take it away, Amaya.
Perfect, great. I can't see anybody and I cannot see the chat while I'm presenting, but you know,
I've got you.
I've got you. That, or we can, you know, you can save the questions for the end. It's fine. I think that we have about like 5 or 10 minutes at the end that we can cover some questions.
Yes.
All right, perfect. Okay, so I apologize. This is a talk that I was going to give at Cranberry School, and many of you heard that I got food poisoning, so I was there but I couldn't give the talk. And then I was supposed to give this talk on another brown bag before, but unfortunately my mom got very sick and I had to go down to Chile to help her. And so the third time is the charm, and here we are to present some of the research that has been done by Pedro Rojas-Barro, who is one of my PhD students, and also, of course, Beth Ann Workmaster. Many of you know Beth and that she works in my lab, and so they have been instrumental in getting this research done. I'm just presenting it, but really all of this work has been done by Pedro as part of his PhD dissertation. So I'm going to be talking today about water and nutrient translocations to berry during fruit development, and some very interesting things that Pedro has discovered that we didn't know about cranberries and how they develop, and some interesting things that happen during the growing season. So I hope that this is going to have, this is going to be, something exciting for you. Okay. So first, just, to give like a very brief general introduction, we all know that when fruit is developing, what really, drives and helps that development is the movement of water and nutrients and sugars into these developing berries, and how the timing of the movement of these different components are going to affect fruit size. And a lot of this is kind of like driven by or governed by, weather. And we've talked many times about how, weather and temperatures and all the things affect the movement of water and nutrient and sugars into the developing fruit. And that's why we always have questions like, well, what happened this year that we had like really like small fruit and we didn't do anything different? And we talk about, how temperature affects all of this physiology and the production of sugars and how these are moved into the fruit. So that is very general. But when we think about it specifically, when we try to divide how is water and nutrients getting into the fruit versus how is the sugar is producing the leaves through photosynthesis moving into the fruit. One thing that we know very well is that within the vascular system, we have sort of a separation of functions. So the xylem are those pipes within, if you think about, the uprights and all the way to the pedicels that are attached to the fruit. The xylem is the one in charge of moving the water up. The nutrients are uptaken by the roots, while the phloem, which is another set of pipes, is moving on the opposite direction from the leaves, all the sugars that are produced into the fruit. But one of the things that we really never have explored is when is the timing of those nutrients moving into the fruit? And this is a question that I get asked a lot because people think about like, well, when is the best time to fertilize? How long should we fertilize? And, we always talk about, nitrogen and when we need to fertilize with nitrogen. But one of the things that I've said many times is nitrogen is really important for the leaves because nitrogen is part of the chlorophyll molecule, and the chlorophyll molecule is in charge of producing photosynthesis and sugars, and those sugars are afterwards going to help with fruit growth. But there's a bunch of other nutrients that go directly into the fruit and do accumulate into the fruit. Some of them, like potassium, for example, we always talk about potassium and fruit growth and fruit development. But we really don't know when those nutrients are being translocated into the fruit. And for that matter, we also don't know much about the water. How much water do those fruits need? How much water, how much do they transpire? So we set up this study to kind of look a little bit about that. And I just have this graph here, and I want to show you a little bit, some dynamics, how things happen. So you can see, here at the very base, you can see we have, water and nutrients that are being taken up by the roots from the soil solution, and they're moved into the fruits and the leaves by a gradient of transpiration. So there's a water potential gradient in which, the water potential is much more negative because the leaves are transpiring. So basically they're just sucking all this water that are coming out of the stomata up here in the leaves, and that is what is driving the movement of this water through. And you can see here this microscopy image that I have through the xylem, which are those vessels here. They're moving this water through that transpiration gradient. And the opposite, what I just explained, when the leaves produce photosynthesis and they produce sugars, those sugars are moved into that fruit through the phloem. But given that this talk is mostly about water and nutrient, I want you to remember how transpiration is really the force that is pulling that water that comes with nutrients into the leaves, but also into the fruits. So transpiration is very important for the movement of water and nutrients. And so what we set up to do is we really wanted to try to understand, if we look during the growing season at fruit development, try to figure out what are some of the changes that are happening in the fruit and also in the pedicel. So that is, the little stem in between. You can see here in this first picture, the pedicel is very clear. So it's that little stem that in between the upright and the berry. What kind of changes happen during fruit development and how those might affect nutrient movement? And since we know that the nutrients and the water are moved through transpiration, we did a lot of studies and a lot of looking into what happens with transpiration, what happens with those fruit, how much they transpire, and trying to infer how that affects when those nutrients are mobilized into the fruit. And so what we did 10 collections. And I said, again, what really Pedro did, he did 10 different collections during the growing season. We started from 1 week after full bloom and went all the way until harvest. And that in this case, we collected the fruits, and also we collected pedicels. We mostly work with Stevens because, as you all know, and I've said this many, many times, it's so hard to do this work. It's so time consuming and so intensive that we just can't do it for all the cultivars. And in this kind of study, we would assume, because it's mostly anatomy, that we have a lot of similarities across the cultivars. There might be some slight difference, but a lot of what I'm talking about today, I think, is generalizable to all of the cultivars. We did this work at the Cranberry Research Station, and we're very, very thankful for Wade and Max for always facilitating and helping us, and also Beth in her role there for helping us to do this kind of work there. And we did the study in 2 years, in 2022 and 2023. And so let me show you a little bit of some of the results that we got. So the first thing we looked into was just, general fruit development. What happens with the fruit? How does the fruit grow? And so what you can see here on the right axis, we have fresh weight of berries, and then in grams, and then on the axis on the right side, we have the dry weight. Okay, so we took that fruit, we dry it completely, and then we took that weight. And what you can see here is like we have an increase, obviously, in fruit size, a little bit more steep at the very beginning, and then it kind of like plateaus as we are approaching harvest, which is what we know. And then the dry weight sort of like follows a very similar trend. It's just like lower values, because a lot of these is basically, it's just water in the tissue. So you can see they start with very little water. It's just mostly dry matter, and then as the fruit develops, there's quite a bit of water that accumulates there. And I have some pictures here of what does it mean, like July 15, where were fruits there? Well, about like a little bit pea size, or a little bit smaller than that. Well, August 15, you have, fruits are almost fully developed, and we start to see some flush. And then here by, mid-September, early September is when we harvest. So this is, just, fruit development, how fruit are gaining weight, are gaining, the growing. And here's the curve. As we were doing these things, here's the curve for potassium. And so here you can see, the accumulation of potassium in the fruit. And so very similar to the weight and the fresh weight of the berries, you can see that there's quite a bit of increase in the absorption and the accumulation of potassium in the fruit at the very beginning. Whoops, sorry. Oh my God, messing it up, sorry. There's quite a bit of gain here from July 11 all the way to about like August 15 of potassium accumulating in the fruit. But then you can see very similar that at the very end, you can see sort of this like plateau. There's no more potassium accumulating in the fruit. And potassium is one of those nutrients that is accumulating in the fruit through the water. As water is getting in the fruit, driven by transpiration, a lot of that potassium is also getting in the fruit. And so, not all nutrients get in the fruit in the same way, but this is for potassium. And I just put this line here, just showing you like this different, like this kind of like break in which we have like quite a bit of accumulation at the very beginning. And then again, we see sort of at the very end of this season, this plateau, and that no more potassium seems to be getting in the fruit by that time. Here, there's a bunch of different nutrients. So very similar to potassium, it's just this graph contains calcium in black, magnesium in orange, and then phosphorus in green. And so what you can see here is, again, we see that same trend for magnesium and for calcium, that we have, an increase in the accumulation at the very beginning of fruit development, but then that just plateau and the concentration is the same. In the case of phosphorus, you see that there's a little bit, it continues to increase the amount of phosphorus in the fruit. And one of the reasons for this is that phosphorus is also, linked to many sugars. So you can get phosphorus accumulation into the fruit just because carbohydrates are still accumulating into the fruit. And those are not getting into the fruit through the xylem, but through the phloem. So this is the other set of pipes that are, in those pedicels that are bringing the sugars from the leaves through photosynthesis into the fruit. So these are two different, highways, one that is bringing just the water and another one the sugars. So in the case of phosphorus, it seems that we have maybe at the very beginning of the season, there might be some phosphorus that is coming from the soil through water and the xylem, and then maybe in this other end of the season is coming through sugars and through the phloem. Okay. So again, we still see this kind of timing in which the fruits are about almost full size, and we're starting to develop that. We're starting to see that change in color from green to sort of like yellowish, and we start to see the first blush. That's when we start seeing that a lot of nutrients, such as like the potassium, the calcium, and also in this case, the magnesium, seem to be, there seems to be no more accumulation in the fruit itself. So one interesting nutrient that had a completely different type of curve when we were looking at the accumulation in the fruit was the boron. And so we have this, bell shape on boron that is very different, and we try to figure out, okay, what would this be happening with boron? And boron is a micronutrient. The plants need it in very low quantity, but it's an essential nutrient. It's very important for pollination. So maybe not incredibly important for in sense of like, fruit development, though it does play an important role when it comes to cell wall and firmness, because a lot of the boron, some of the boron is associated with some of the pectins and the hemicellulose that we have in the cell wall that gives a structure to that pulp. So if we have deficiency of boron, we can also have, softer fruit. So in a way, it's important for fruit quality. But what we see in this case is that we see like a steady increase, and then it just really drops the concentration, and then it kind of like plateaus at the very end. So boron, we don't know why is that. We hypothesize and we think that a lot of what's happening here is that some of the, that boron is immobilized and remobilized and moved from the fruit in and out of the fruit. But this is one of those nutrients that have sort of like a different pattern, and we really haven't explored really well why this is happening, but just to show you that not all nutrients behave the same. But the major micronutrients seems to follow that trend that I just explained, in which we increase and then we tend to plateau in mid-season. So as I was saying before, a lot of these nutrients move through the xylem and move through transpiration. So one of the things that we wanted to look into was like, well, is there something maybe happening in the pedicel itself? So is there something happening here as the fruit develops that might be affecting the movement or conditioning the movement of water and nutrient into the fruit? And so besides just looking at the amount of nutrients that we found in the berries, we also did some sampling of those pedicels as the fruit developed, and we did some microscopy work, and we wanted to really look at what is happening with these vessels. And I call them like pipes, because that's what it is, that they're moving this water and nutrient. Is there maybe something like they're stopped functioning? What do we know about it? And is there something that it can tell us about nutrient movement into the fruit that could inform us about how to change some of our fertilization practices? So what I want to show you with this graph is showing the xylem vessel density. So how many of those vessels do we have in those pedicels as the fruit develops? So when we started, here at this size. Fruits were very, very small. Actually, we started at pinhead. And we would take those pedicels, we would make cuts on them and look in the microscope and try to figure out what's the density of these xylem vessels that are moving the water and the nutrient into the fruit. And what we find out is that as the fruit develop, we see an increase in the density. So we see more and more and more of those vessels being developed. But then again, you know, at this kind of critical stage in which we start seeing, you know, color development and this change between the green fruit to kind of more like yellowish fruit, and then the blush development, we started seeing that this sort of plateau, and here we see sort of a bit of a decline. So the density increased, but then again, the same trend sort of plateau. But then when we looked at the hydraulic mean diameter, this basically is trying to calculate and estimate what is like the overall like potential of moving water. It's not only, you know, the density and the number of those vessels, but what is their potential of how much water they can actually move through the pedicel. We see that as the fruit develops, there's the hydraulic diameter, the amount of diameter they have to move water starts decreasing. There's less and less water that can actually move through the pedicel into the fruit. Okay, so that was something really interesting. It seems like the flow of water is much bigger at the very beginning in fruit development, and then it really kind of like decreases as the fruit grows, and it steadily decreases all the way until harvest. So less and less water is getting in, is moving from the pedicel into the fruit. And so we're like, oh, well, should we also start looking at whether, like, not, these are all anatomical like measurements. So like we knew that there were higher density of vessels, but that overall those vessels were getting smaller and smaller and smaller. So like overall the capacity of moving water was less as the fruit was getting ready to harvest. But another thing that we really wanted to look into is like, well, are they really functional? Are they actually moving any water and nutrients into the fruit? Because we saw that plateau, and so that means like, well, maybe there's like nothing happening. That maybe that pedicel is just not functional anymore when it comes to the xylem, so the vessels that are moving water and nutrients. So what Pedro did, which is very smart, is he decided to use, what if we use a dye to figure out by tracking that color whether the water is getting into the fruit as it develops. And so what he did is he sampled a bunch of uprights with a bunch of fruit either attached to the pedicels, as you can see there, or he also detached them, as you can see in the picture in the bottom, and he put them in a little, you know, container that had red dye. And we put them into conditions in which, you know, you would have a lot of wind that would mimic transpiration. So we tried to create that water vapor deficiency so that it would simulate exactly how the plants are in the field, in which the air is drier, so it's pushing that water out of the leaves. So we try to simulate that in the lab by putting these little uprights in this red dye solution, and then also created the conditions so the leaves and the fruit will continue to transpire and try to move water into the fruit. So here's a really good example. You can see here, we did this for 30 minutes, and you can see at the very beginning when we put the fruit and the uprights in the little jars, and then what happened after 30 minutes, you can see when the fruit is transpiring and the leaves are transpiring, the dye is moving into the fruit, and you can see how everything turned red. So we did this experiment when the fruit started like very tiny at pinhead, all the way until we harvest. And so what is it that we saw? Well, you can see here in this graph is the percent of fruit that had dye inside. And so we dissected the fruit to look at that. And what we saw is that at the very beginning, all of the fruit was actually getting this red dye inside. But as the fruit developed, and again, I want to show this image of this critical change of color that is happening, this stage in the fruit development, we started seeing that there was less of that dye, and that dye was only getting at the very at the portion of the fruit that is attached to the pedicel. But then as the fruit continued to develop, none of that dye was moving into the fruit, meaning there was no water and nutrients getting at all into the fruit. And so this was like really interesting that we figure out that at some point during fruit development, water and nutrients are just not getting into the fruit anymore. And the critical point seems to be when the fruit reach this, considerable fruit size, but we mostly figure out that it's when the fruit are changing color, so changing from green to this kind of like more whitish, yellowish, and it's the very beginning of the blush and the color developing in the fruit. At that point, when we were analyzing, when I show you those graphs of calcium and of magnesium and of potassium, that's when we started seeing the plateau. And the plateau seems to happen because that xylem stops being functional. And we prove that by showing you that there's no more dye getting into the fruit. And so that shows, it's a really good proof that, water and nutrients are not getting in there. And so here, this curve shows at the stage in which 50% of the fruit will not have dye in any of the portions of the fruit, okay? And so another very interesting thing that we saw as we were doing this test is that we started seeing an abscission zone develop. And so what I mean with an abscission zone, the pedicel started, there was something that was started sort of to detach it from the fruit. And even though we need to really pull those berries to harvest them, there is some sort of separation already happening that might be contributing to the fact that water and nutrients are not getting into the fruit. And this happened in August, and we did a lot of microscopy work. But I think that one of the things that is most evident is that you can see that some of that dye is leaking out of the fruit right here. And so that also shows us that there's some abscission zone that is happening. This is very common on other fruits. We tend not to think about this in cranberries, but if you think like, about like apples and peaches and all that, if you let them ripen the fruit, they will just drop because this is that abscission zone that is happening here. In the case of cranberries, we need to pull the berries out of the vines, but that doesn't mean that there's no abscission zone. And we did show this with this when we looked at this phenomenon of this dye kind of like coming out of the fruit, even though the pedicels were intact. Internally, when we did the cut, we could see that these abscission zones are happening here, and the tissues seem to be rupturing there. So very preliminary conclusions. We have a lot more. I mean, Pedro has a lot more that he has done and will be, providing more information about this. But it seems that water, nutrient, and sugar movement into the fruit definitely declines as the fruit develops. Okay, and that's especially true with the data that we show with the water and the nutrients. By the second week of August, about 50% of the fruit did not take any dye, which means 50% of the fruit that we tested, there was no water moving into the fruit whatsoever. And then also there seems to be some evidence that there's some fruit isolation suggesting that the water and nutrient movement into the fruit stops approximately about 4 weeks before harvest. So that are some of the preliminary conclusions that we got out of this study. And, again, just to acknowledge and thank the funding that we had for this study that has a lot more information in it. So the USDA funding that we got, and also really to thank the station for facilitating the research there and for really helping us do all of this crazy stuff that we do at the station. And so we really want to thank Wade and Max and Beth in her role as the research manager of the station for letting us do this work. And so with that, I hope I didn't go over time, and I'll be happy to answer any questions.
Yeah, we are good to have a couple of questions, and if people want to ask their questions either by asking and unmuting and asking. In the meantime, I just made the note for people following along on the closed captions. Closed captions loves to hear pedestal. It is actually the pedicel, P-E-D-I-C-E-L. If you are if you were baffled by that, I made a
also
note.
I know that my accent doesn't help, but I mean you folks are used to it. So.
No, the closed caption doesn't have a lot of botany knowledge. When I make these updates, there's a lot of botany knowledge that, like, well, it's a good thing we know what's going on. I had made the question and you basically answered it, but in your chart of, for example, boron in milligrams per gram of dry weight means the dry weight of fruit only, or tissue plus fruit. That meant the fruit only, right?
Only fruit.
Yes.
Only
okay.
Fruit. Yeah.
Another question. We've got a lot of, wow, very good information and great topic by Amaya. One somewhat related topic, is there any research planned or interest in developing an evapotranspiration model for cranberries, especially as more and more growers get weather stations and could be gathering data? Any thoughts on that?
No, not that I know of, but I know that there's a new researcher here at UW-Madison, and she works a lot on irrigation, and we connected, and I told her, like, we have a lot of things to do on cranberry, so I'm going to try to bring her in, and she's a specialist on irrigation, and so it would be great to have her, to talk to her, and to maybe just, I've been already bouncing some ideas with her, but I think that would be a great topic. The only thing that I want to make the distinction, just because you brought this up, when you think about evapotranspiration and all of that, we're thinking about foliage and leaves. What I presented here, and I will be presenting more of this project, is mostly related with the fruit. How's the fruit transpiring? And I think that maybe Pedro talked about this a couple of years ago, but we have some data on there, because it's the transpiration that is moving the water. It's just like, basically like when we drink out of a straw. So the end of the straw is that pull from the environment, from the air, and that's what's bringing the water into the leaves, but also into the fruit. But a lot of the data that I presented today is like related to the fruit itself, which is a very different organ when it comes to transpiration than the leaves, with that their main goal is to transpire, which is not the case of the fruit.
Absolutely. And I think I guess I've got a couple more questions already here, and probably time for 2 more after these 2. So if people have questions, go ahead and bring them in. But so are the most important nutrients nitrogen, potassium, and the micronutrient boron in terms of filling fruit?
So there's the fruit is going to have all of the nutrients, okay? But when we think about like macronutrients, those are the nutrients that we talk about extensively about this, the plant needs in really high quantities. That's what they call macronutrients. So we tend to focus on that just because there's more of those components in the fruit itself, okay? And that's what we're looking at. And the reality is that this study started with the idea of trying to look at calcium. We were mostly interested in calcium because, as you all know, my lab has been working a lot in trying to understand like fruit quality and what makes a good fruit to be a sweet and dry cranberry. And calcium is really important for the cell walls and the structure of the pulp of the fruit. And so we were specifically looking at calcium, but since we were doing this, we did the analysis for other nutrients. And so we started figuring out that the pattern was very similar. And so boron is an exception, and I show it there just because, it's a different kind of pattern of movement into the fruit. But boron is also, the fruit and the plants really needs a very, very, very small amount of boron, okay? It's one of those nutrients that you go from deficiency to toxicity in like nothing, like very, very, very small range. So it's an anomaly in the way that it moves into the fruit, but also it's not something that we are constantly fertilizing for. What I think is important, and one of the takeaways for me here is that, and I'm sorry if I sound like a pain, because you've all heard me say this so many times about like, over fertilizing and thinking that fertilizer is going to solve your problem of fruit size, and that we're going to fertilize, we're going to put more potassium because we need to get, because that's going to help us, with fruit size. I really want to show you with this research that fertilization at the end of the season, not even at the end of the season, we're talking about here like mid-fruit development, that fertilization at that point is really not doing anything for you, because I just show you that water and those nutrients are not getting into the fruit at that point. And so when we get to the point in which the fruit starts developing, the flush, and they start to change color, it seems to be a critical stage in which a lot of other things are also happening. And one of the things that are happening is that the water movement and the nutrient movement into the fruit is kind of like minimal. And it looks like the last portion, maybe like the last fourth or even the third portion of development of the fruit is there's mostly sugars coming from the phloem. I only show what was coming from the xylem. So there's sugars coming from the phloem that are getting into the fruit and are helping to continue to develop it. But in terms of fertility, that we're thinking we're going to put more nutrients into the fruit, that is really not happening. So when you're doing some of those fertilizations in August and late August, those nutrients are definitely going into the plant, and they're going into the leaves and into the other structures of the plant. But don't fool yourself, they're not going into the fruit, and they're not necessarily going to help you to increase fruit size at that stage. And I know it's very frustrating, and we talked about this because there's so many other factors that are affecting this. But I want to add this piece of information just to let you know that when you are going to do those fertilizations, just keep in mind that those nutrients are not going into your fruit.
And I'm gonna, we are like 1 minute over, but I'm gonna smush two similar questions into one question and let you handle that, and then we'll move along and let you head off to the thesis defense. But what is the take home? Is there a fertilizer application that should go on at a specific time to maximize fruit size? And then the second question that's kind of a follow-up, so should fertilization be finished at the beginning of August? You kind of addressed that with the well, after fruit, maybe there are things going into the tissues, but do you have a suggested specific timing for fertilization to maximize fruit size? And then after that, it shouldn't be considered for fruit anymore?
Well, if you look at the graph and like everything that was happening, you could see like at the very beginning of fruit development, everything was just going really fast. So the timing of fertilization, you know, when we're thinking about fruit, it has to be at the very beginning of the fruit development. So when those fruits are setting and they're developing their pinhead all the way until pea size, this is the period of really, if we want to get those nutrients into that fruit, that's the period. I think that if you're just moving now, like, you know, mid-August or after we go, like, I don't want to give specific dates because, you know, we know that we have different timings, but when we start seeing that those fruits are changing, that slight change of color, it really means that anything that is going in to the fruit through the xylem is really declining. So there's no point of doing. We can fertilize, we can still fertilize, but just remember those nutrients are going to go into, you know, the roots, the stems, the uprights, the foliage, but not into the fruit. And so that's my recommendation.
Thank you. This is, I'm going to go ahead and switch to my slides again, but thank you so much, Amaya. Thank you to Pedro, and obviously Max and Wade at the research station, as well as Beth. And then also just what exciting timing this is. I know literally all of the nitrogen optimization pilot project programs that we have going on are focused on nitrogen timing and being able to say, with this, you know, armed with this new fundamental research knowledge, we're going to have an even better time understanding what results we see from that. Is that letting me share? How about share screen and PowerPoint? Awesome. And I am going to have Pam be our pinned speaker here for a minute. You folks see my screen, I believe, and I just have to make it be larger. There's closed captions on top of it. Cool. Sorry, Pam, go ahead and unmute yourself, and thanks for joining us. But again, you know, a huge thank you to Amaya. If you have more questions, feel free to follow up by email, by text, however you like, and we'll be sure to keep those answers coming. And then Pam, I believe we can see my screen now, so you should be good to take it away.
Yep, we can see it. Yeah, and I just sent a bunch of pictures to her right before this, so thank you for throwing them up there last minute. So this one is prompting me to talk about pollination. A lot of growers are like, man, my bees aren't working. What's going on this year? And we're out there doing the bee surveys with, we're working with Claudio and Hannah Gaines on collecting bee surveys. And this year, we can tell you absolutely we're seeing less activity than last year. And then we're not done yet. There's still blossoms out there. We're still going to do more surveys. But Hannah said in apples, like their work is done, and literally they had half as many visits in apples this year than last year. So what you're seeing is happening, but we'll just see what happens after. We'll see what sets. you can, I don't know what picture you have next. Okay. All right, this one is false blossom. This time of the year, it's really easy to see. It has those like witch's broom sticking up blossoms. And again, this is spread by the blunt nose leafhopper, we believe. And I wouldn't say there's a lot more popping up. Maybe like 1 or 2 properties. We found like a few more little areas, but it's kind of similar to last year. Okay, this is the one to kind of follow up Amaya's presentation on fertilizer. This year this happened probably like last week when it was raining a lot. And what happened is the grower put his fertilizer out and it was supposed to rain. So he didn't water it in right away. But again, you guys, if even if you have a little dew or something and that fertilizer sticks to your blossoms, it will burn it. So even if it's going to rain, just try to like get it rinsed off and then let the rain incorporate it later. But if you don't, then this will happen. And we're starting to see what is probably going to be early rot. If you look really close, you can see the little black pycnidia on this. This is like a 2-year-old planting. So it's out there. And I think I've been telling my guys, if you're on the fence, this is probably going to be a year of pathogens just because of the hot temperatures. And then it's spreading with water and it's raining so much. So that is probably going to be early rot. Talking about the rain, a lot of water in the ditches. Growers are just trying to get rid of the water. I think you can keep flipping because I think the next few are just either ditching. Yep, get in there, ditch it, cut the grass, adjust your flood ditches around, just try to get the water to move off your beds. I think the next one might be, yeah, if you look close, the blossoms, if they are wet too long, you will lose them. So this guy lost probably about a foot and a half of blossoms all along the edge. So.
I had a question come in. Do we have any idea as to why bee activity is down? Does it have to do with the weather variation from last year to this year?
Yeah, so that would be my guess. I asked Hannah what she thought too. And so like last week, for example, there were a lot of clouds and it rained. And the best environment for bees is when you can see your shadow. So if you could see your shadow and it's like a gentle breeze or no breeze, then we see a lot more bees in our surveys. So our surveys, if you're not familiar, we use the Weebee app and we stand out there for 5 minutes and observe the bees. And if it's really windy, they're just not out there. And if it's cloudy and if it's rainy or wet, they're just not out there. Hannah is also collecting pollen from the different honeybee hives. So she's going to be analyzing that to see if they are working another crop, like a non-crop plant or something that maybe this year is blooming or better because of the rain. I don't know. But that would be my guess is the weather. She says there are variations in bee colonies from year to year, but typically not honeybee because they're I can see this question, the native bees. So bumblebees, this is just purely from doing the Weebee observations. So bumblebees in my observations tend to not be as picky. So when it is cloudy, we'll sometimes see more bumblebee activity than honeybee. There are growers that have the bumblebee hives and honeybee hives. And if they do have both, we tend to see more bumblebees when it's cloudy. The native bees and like wild bees, I would say it's pretty strong. I mean, I don't have the data yet from all of our surveys, but we're doing like 500 surveys this year. So that's 500 stops in the cranberries and then whatever Hannah and her crew is doing. So she should have some pretty interesting data for us at the end of this pollination.
And Hannah did commit to giving us an article in the August, like the first August Cranberry Crop Management Journal that's going to have all the details of who visited where and what results in cranberry this year. So looking forward to reading that.
Yeah, and one of the observations we're doing is marsh edge. So along the tree line, along the reservoirs, along where maybe you have a pollinator garden versus the center of the marsh, which is just that in the middle. And just from my observations, I do see more native bee activity on the edge, which is probably assumed to be the case everywhere.
I think let's keep having questions come in. I will show this. Anna Maria has her screen cap or take that QR code to take her survey. It is intended for cranberry growers in the US and in Canada. So no need if you're Canadian or if you're Massachusettsian or if you're Oregonian, you may still take that survey and contribute. So I just figured while we take questions, I could show that. Other questions coming in for Pam? I guess I can still continue to give the, if you do see false blossom, as she noted, this is a good time of year for noticing that. I believe that Leslie and her team are still taking volunteers that would be anonymous, but being able to participate in research, whether you want advice in roguing that out or whether you would like to have it be participating in research, reach out to Leslie, which is Leslie, laholland@wisc.edu. Or you can write to me and I'll connect you. But if you go direct to Leslie, that can stay anonymous too. Yeah, do we have concern about pollen being washed off by the rain?
I don't know if Amaya wants to answer that or not, but
Amaya,
that is definitely something that a lot of growers ask.
I'm not, an expert on this. We should have an entomologist here to talk about this, but I wouldn't think so, because the stigma, around the stigma, it's really sticky. So once the pollen lands there, it just stays with this kind of like sticky substance around it. So I wouldn't be concerned that it's gonna wash off. I think the biggest concern and why we see less pollination is just because we see the pollinators are just less activity. But once it lands there, and the conditions are adequate, you will have the germination of that pollen really fast, and fertilization will happen very fast. So I wouldn't be concerned about the pollen washing off, but I haven't done the research and it's not my area of expertise. So that's what I think.
Thanks. Yeah, I'm not sure either. I know just when we're out there doing our percents, we always kind of brush the tops to see what petals fall. And usually we see like all the little smoke, the little pollen just flying around. But when it is wet and when there is dew, obviously that's not flying around. So I'm not sure. I see that Lindsay's on here. I'm kind of curious how the East Coast is looking for disease and if you got as much rain as we did. And I don't know if you want to comment on anything.
Jeez, putting me on the spot, Pam.
Thank you.
We have not had much rain at all. Unfortunately, we're pretty dry. We've gotten a few pretty timely rainstorms, so I'm thankful for that. But we've had kind of the opposite of you guys this year, where we've had pretty good pollination weather. But that being said, we've been really, really hot. So, upwards of 90 almost every day during bloom. So 90 Fahrenheit, I should say. Yeah, but we'll see. So we moved through bloom, or we're moving through bloom pretty quickly.
Yeah, that's another comment I will make, is that the bloom is not progressing super fast. We were doing our bloom calculations, and during that time was pretty warm, and we were getting probably like the 10% gain per day. But now as we're moving out of bloom and we're trying to time the fruitworm sprays, I would say more like the 5%. Like it's hanging on, which hopefully will help us later when we get some good weather. But
I need to make a verbal correction. I had put up our next slide here saying that our July brown bag is going to be back to the normal fifth Friday, or the normal third Friday of July 15th. And then also I had an error up. The summer field day is correctly going to be Wednesday, August 14th. So that's Wednesday, August 14th at Cutler Cranberry in City Point. And I just wanted to make sure that I verbally said the correct thing so that no one has the wrong thing stick in their mind. Had another question, may the pollen die because of the hotter temperature? Probably referring to New Jersey's.
It doesn't die, but it would desiccate.
Might desiccate, okay.
And so as soon as you get the pollen out and it's really, really hot and dry, it just desiccates and then you don't have any germination. And also the stigmas of the flowers will desiccate, and they, you know, the life of the flower itself will be a lot shorter, and the stigma can dry. And then once the stigma, which is the top of the bottle in the female structure of the flower where the pollen is to land, if that dries, then it doesn't matter if you have a lot of pollen there, the pollen is not going to be able to germinate and get all the way into the ovary to fertilize those ovules to have the fruit.
Is that once it begins to get dry, if you get rain and it rehydrates, is it okay, or is it an irreversible process?
For the stigma, irreversible. Yeah, for irreversible. The pollen, I don't know. Again, I think that's a good question for an entomologist.
Okay, yeah, fair point. I guess, and Amaya, since you're still here, I want to kind of bounce back to some of your earlier, you and Pedro's research. When you were assessing the fruit growth with the dye, did you notice dye tracing up into the tissue in the latter half of August, or is that not a dye that gets uptaked by tissue, and so we're going to have to do something else to do that kind of research?
No, the dye always moves into the leaves, always, because the leaf will transpire always.
Oh,
so
okay.
we're talking about, because so what happens? So the leaves have so many stomata, which are those little openings in the leaves that allows that water to go from the tissue into the air. So that's what really drives this water vapor deficiency gradient, that is, you know, pulling the water from the soil all the way into the leaves, into the air. And so the leaves are transpiring, because what are they trying to do? The same thing that we do when we transpire. We're trying to, when that water evaporates out of our bodies, taking out the heat out of the body. So the leaves are transpiring to cool the plants. And that happens all season long. That is, you know, one of the major functions of the leaves is the photosynthesis, but transpiration and controlling the temperature is really important. Now, the fruit has a lot less stomata, very few stomata. And most of the stomata in cranberries are actually not functioning. So the transpiration of the fruit is, it's, you know, 100th of a time of what a leaf can do. So in the case of the dye that is mixed with the water, so that dye with water is basically representative of the water that's in the soil. As the fruit develops, the water continues to go into the leaves without any problem. What happens is that the fruit stops transpiring, and the pedicel starts breaking down. And so that's why no water and nutrients are getting into the fruit anymore. So we start seeing that decline on those graphs that I showed you.
Okay, awesome. I have another question that I know a lot of people are going to say, yeah, we know that already, but can you give us the basic, I know you kind of referred to them both as pipes, but what's the basic breakdown between xylem and phloem?
Yeah, so as I said, the function of the xylem is to move water and nutrients from the roots. So the roots take up the water and the nutrients, and then those are directed into the xylem, and the xylem transport that everywhere in the plant, including the fruit. The phloem, that is, you know, another set of basically vessels that are all over the plant. what they transport instead of water and nutrients is like they take all the sugars that are being made in the leaves from photosynthesis and redistributes all those sugars, all those carbohydrates to every part of the plant that needs them. So within the same leaves, it takes them to the roots, it takes them to the stems, and it obviously takes those sugars and carbohydrates to the fruit. So those are two, and so the pedicels have both xylem and phloem.
Okay, and so the xylem is water and nutrient pipes, and the phloem is sugar pipes, basically. Got a question from Jocelyn here. When the plant transpires and before the abscission zone is made, so when the pedicel is still functioning, can a fruit be sucked by the leaves? Can the leaves say, hey, I want more, and maybe the roots are dry? Can the leaves slurp moisture from the fruit?
There's no moisture going from, there's no water being pulled from the fruit back into the upright. What can happen is that since this is a unit in which you have leaves and you have the fruit, the pull of the leaves is so much more stronger than the fruit, that when we had those tests with the fruit attached to the upright, we saw that the fruit stopped moving any dye in much earlier than when we did the same test and we detached the berry and we only put the berry in the solution with the dye. So what's happening there is such a competition, so strong the competition from the leaves, that, and this is what happens in reality in the field, because I mean we don't have detached fruit there. The leaves are just moving all the water and the nutrients in that direction, and then very, very little is going into the fruit as the fruit continues to develop. Because I just want to remind you, it's not that it never moves in. Like the graph really shows at the very beginning, there's quite a bit of movement of water and nutrients. It's just that as the fruit develops, that really just stops and plateaus, and then there's no more.
Awesome. I really appreciate all the questions coming in. Appreciate Pam for her presentation today and Amaya for her presentation today. We've still got time for 2 to 3 more questions. So good, we've got one. After fruit set, what causes a plant to abort fruit?
Well, multiple things. One of them could be, and this is very appropriate with some of the information that Pam was sharing. It might just be that there was really no fertilization. There's no seeds, there's no embryo being developed there, and so the development of the fruit. I'm sorry, Amaya is going to get into her plant physiology lecture here.
We need.
How does the fruit develop? The fruit develops because when the pollen germinates and those ovules are being fertilized, and we have the beginning of the formation of the embryo in the seed, that kind of like triggers this production of hormones. And that stimulates that the ovaries, the wall of the ovaries starts developing into the fruit, okay? Sometimes you can get that stimulation and a little bit of development and enlargement of the walls of the ovaries. And so what start to look like a fruit is going to develop just by having pollen land in the stigma, but not necessarily for fertilization. So this happens a lot in many, many fruits. So you see that initial, kind of like enlargement and started like a production of fruit, but then it stops because there's really no embryo that is continuing to produce those hormones. It's just the initial stimulation from the pollen. But then if fertilization never happened and there's no embryo there, it would die. And so we see that that really happens a lot. This is also the process by which we create seedless fruit. So when you eat some oranges, they're seedless. In this case, those fruits are able to develop even when there's no embryo because there's such a stimulation, and you can do it with application of exogenous hormones, that it will trigger the enlargement of the ovaries. So that's how we do, seedless fruit. But here, for your point, what's happening is that might be that you got some pollen on the stigma of the flowers, and that is stimulated a little bit the initial enlargement of the walls of the ovary. But then what is needed to continue to get a full development of fruit is that embryo, and if it's not there, then the fruit would just abort. I don't think, or maybe I haven't dig really well in the literature or don't remember anymore, but I don't think, I want to remember that somebody did a study in which they specifically kind of like, they were doing thinning, so they were removing some of the berries to see how much of an impact that would have in developing more fruit, thinking that, oh, maybe the upright self-regulate. But I don't think that that is something that happens on cranberry. Well, in apples, we know very well something that we call the June drop, in which you see a lot of fruits that just drop out of your apple trees when they're still tiny. And that's because the tree is self-regulating, because it can take to completion all of the apples in the trees. I've never seen that on cranberries. It's just, you just see that they just stop developing. And I don't know if that's because it's self-regulating and stops developing certain fruit, or it's mostly due because they were not properly pollinated, which is my gut feeling that that's really what's happening. Sorry for the very long answer.
No, we appreciate it. We have made it to 1 o'clock, so I want to just take one last minute to thank Amaya, thank Pam, thank the Growers Association, along with UW-Madison for supporting our virtual brown bag series, as always. I'm going to end the recording here after thanking everyone. Oh, that was the screen share pause. How about the recording pause?